welcome back. I hope you guys had a good time in the networking event. And we're back for the second half today. We're really excited to be joined by several excellent speakers in this jam-packed session. It's session 3, Technical Advancements in Clinical Machine Learning, What's New in 2020. This session will be moderated by my colleague, Akshay Chaudhari, who's an instructor in radiology and the Associate Director of Research and Education at our Center for Artificial Intelligence and Medicine Imaging. Welcome, Akshay, and take it away.
Thanks, Matt. I'm really looking forward to this session. We have a great lineup of speakers ahead. And really quickly, just the motivation behind this session was the fact that medicine and medical imaging, they face very unique challenges that we may not face in computer science. You know, for example, medical imaging is not simply a grayscale ImageNet. There's unique insights and challenges and optimization criteria that we have to face. So we have a great lineup of speakers. I'll be focusing on combining some of the clinical insights as well as technical advances, and trying to speak that bilingual language that Daphne had talked about today. So I'm really looking forward to this. And just a logistical note for the audience, please post your questions for the speakers in the chat. I'll be monitoring that and be using that for our panel session at the end. So with that, I'm very excited to introduce our first speaker of the day, who will be Jeremy Howard. Jeremy is a founding researcher at fast.ai, which is a research institute dedicated to making deep learning more accessible. He's faculty at the University of San Francisco and chief scientist at doc.ai and platform.ai. Previously, Jeremy was a founding CEO of Analytic, as well as the president and the chief scientist of the data science platform that we all know and love, Kaggle. Jeremy is an entrepreneur, business strategist, developer, and educator. And today he'll be speaking to us on applying self-supervised transfer learning to medical vision and NLP tasks. Jeremy, please take it away.
Thanks very much. I'm from the University of San Francisco. I think in the program, I just noticed this morning, it actually said UCSF. I'm not from UCSF, I'm from USF, just down the road from UCSF. I'm from the AI and medical research group called AIMI there. And the work I'm showing today, SOLC, which is perhaps the world's top research institute for life sciences. It's been very exciting so the way AIMI, our research lab, works is that we work in a very cross-disciplinary way. We combine data science and AI experts in our group with medical and life sciences experts at academic research and medical centers, such as Harvard, Stanford, UCSF, and Salk. The work that I'm presenting has been done over the last, it's really been over the last 3 years, in fact. The first year of research was largely done at my research lab at fast.ai, and then it was further developed by AIMI and Salk last year. And then I'm going to be presenting some additional development that was then done this year by this group of people. Yuri Man has been involved throughout this process. And so it's been a fantastic cross-disciplinary team over quite a long period of time at this point. The problem that we endeavored to solve together was the problem of microscopy. On the left here, you can see how we can scan quite quickly at a 8 nanometer pixel size. This is with an electron microscope that costs about $1.5 million. The problem with that fast scanning is, as you can see, it's quite low resolution. But if we want to go down to a 2 nanometer pixel resolution, you can see it scans very, very slowly. There's this problem in microscopy that you can generally get either high resolution or speed, but not both. Here's another way places as a problem. You scan at high resolution, you can see here it actually falls apart. So the actual tissue that you're trying to analyze, because of the large amount of basically photons that are hitting it, it actually literally falls apart. I'll show you that again. So it starts out with a nice high resolution picture, and then suddenly it's looking like trash. On the other hand, you can use an 8 nanometer or lower resolution, and you'll get something that actually you can see what's going on. But it's pretty hard to see what's going on in the detail. So for doing stuff like connectomics, mapping the structure of the brain, which is what the Salk folks are trying to do, you really want both. You need high speed, and high accuracy. So we decided to get together with them to see if we can apply some of the ideas that we had developed at USF and fast.ai around super resolution. And so we actually taught a new approach to super resolution in the fast.ai course about 2.5 or 3 years ago. And you can see the result at the top. Very pixelated bird image ends up looking pretty good. And this medium resolution cat image, if you have a high resolution display, I don't know if Zoom can show you this, but it ends up looking incredibly crisp. One of the nice things about this algorithm, like pretty much everything we do, is it runs on a single GPU within a matter of minutes or maybe an hour or two. So it's fast and not at all resource intensive. So we thought, could we apply something like this to medical imaging? One of our students actually showed how well you can apply the basic approach to the extent that actually he developed further on top of our idea to not just do super resolution, but colorization. And that ended up becoming a commercial product at MyHeritage that is now used by millions of people. And you can see how black and white photos become color photos through using almost the exact technique I'll be talking about today, with some clever tricks that have been added by Jason and Dana, who are the fast.ai students who developed this into a very successful business. So the basic idea here is image generation. It's taking an image in and generating some other image out, where the out image is different in some way, higher resolution, color, whatever. So the basic approach that we teach for this in fast.ai is to start with a photo, an image, whatever, and then what we call crapify it. So make it into a really awful version of that. And the way we've done it in this case is we've added really low quality JPEG compression. We've added some text on top of it, and we've blurred the whole thing, and then we've rotated it. Actually, sorry, I didn't rotate it in this case. And so it's very easy to create training data in this way, because you can take any high quality data you have and synthetically create low quality versions. And then you can train a convolutional neural net based approach to go in the opposite direction. To say, okay, given this crappy input, can you recreate the high quality original? So that's why we call it decrapification, which, believe it or not, is the term of art, which we used in the paper and in the software. So it's a nice general technique, because if you want to, for example, do colorization, then you can start with a high quality original color. You can turn it into a black and white, and then train a model to go in the opposite direction. So there's all kinds of things you can do with decrapification. So the basic technique we used for microscopy was to say, okay, let's create our crappy images, generate a estimated high quality version. So that's our prediction. And then take the original high res image and then compare them using pixel mean squared error. And when you train a model in this way, so you're basically training the generator, the actual generator architecture that we use, in fact, pretty much everything in this approach we use comes out of the fast.ai library. There's a paper about these approaches showing how fast.ai works if you're interested. But one of the many things in fast.ai is what we call a dynamic UNet. For anybody doing medical imaging, you're probably familiar with the UNet. The fast.ai version adds lots and lots of tricks. fastAI is an open source piece of software with an academic paper about it. And these are some of the tricks that we use in the library to get much better results than a traditional UNet. But the basic idea would be very familiar to anybody who's worked with UNets before, but you just find you get some rather better results. So using that approach of decrapification and pixel MSE, loss, the dynamic UNet, and so forth, this was basically where we were last year. So as of about a year ago, we started with the low resolution acquisition. And if you use a simple bilinear interpolation to try to upscale it, then you get well over 40% false negative rate when the actual experts try to do basically find the synaptic vesicles in the image. So you can see the dots here are the red dots are the ones where they messed up, basically. Whereas in the version with this algorithm, where instead of using bilinear interpolation, we use this algorithm, you can see it's much more clear. You can see the synaptic vesicles much better. And most importantly, in the actual downstream task, when the experts try to find the synaptic vesicles, in this upscaled version using the neural net, there's only about 13% false negative rate. And of course, in the original high-res acquisition, you get a better result still. So as of about a year ago, we were getting really great results. One of the concerns, though, was that the upscaled version of this algorithm was pretty blurry. And we were pretty sure we knew why. We were pretty sure that it was because we were using mean squared error as the loss function when we were trying to generate the images in this unit. So the idea we decided to go for was to try and adapt the perceptual losses approach used in style transfer that actually came out of Stanford a few years ago. And it's again something we teach in the Fast AI course. And the basic idea is that you take the image that you're trying to match to, and you pass it through some pre-trained network, and you pull out the features at some layer. And then rather than doing mean squared error against pixels, you're doing mean squared or L1 error against the features of some layer. But we have a problem. We don't really have pre-trained networks in medical imaging that you can just throw at a problem. We certainly didn't for this kind of microscopy task. So what do you do for the loss network? Well, we borrowed an idea from the self-supervised learning literature, which is we basically invented a dummy task, a task which wasn't important of itself, but if a model was able to do that task well, it would be required to create high quality, useful semantic features. So the dummy task we selected, again, comes from the self-supervised learning literature, is what's called in-painting. And in-painting is where you take your original microscopy, in this case image, and you delete some rectangular sections from it. And so we developed this patchmaker simple algorithm, which would create a few random rectangles of different shapes and sizes in different places and delete parts of a picture. And so then we trained a CNN, actually again a unit, to try to predict what were the contents of the thing that was deleted. So in order to try to guess what was in this box, you need to, the model needs to get a pretty good understanding of what this kind of microscopy looks like. So this model, which can do this in-painting, becomes a model with some pretty useful semantic features. So this kind of approach has been used before for self-supervised learning, but we used it in a very different way. We took this in-painting model that we developed, and then we replaced the MSE loss with perceptual loss, but the perceptual loss was against a pre-trained model that was trained for the in-painting task. And so that means that you can then actually try and make sure that the model that's being built has appropriate textures, appropriate features, something that we would expect to work a lot better. And indeed, it did. Here is a few different zoomed in levels versions of the mean squared error deep learning upsampling approach. And as you can see, you're seeing this kind of this blurriness going on. But with this new feature loss based approach, as you can see, you get an extraordinarily high level of detail. Particularly once you really zoom right in, it's quite amazing how it's actually able to recreate the original image extraordinarily closely. So we're really thrilled at where we've got to. We think that this is not just a useful idea for microscopy, but this is a useful computer vision idea in general is particularly useful for medical imaging, where there is unfortunately still a real shortage of pre-trained models. And so with this approach, you can build generative models. And in fact, there's all kinds of things. You could do all kinds of self-supervised approaches and so forth by modifying this basic idea. It really helps get around the lack of pre-trained model issue in medical imaging and allows us to do things which really weren't possible before. I hope that was useful. Thanks very much.
Thank you for that very nice talk. It seems like with these deep learning techniques, we're trying to have our cake and eat it too. So looking forward to see where
I hope so.
with that. Yeah. Well, moving along with the session, our next speaker that will be joining us is Dr. Sharmila Majumdar, who is currently the Margaret Hart Surbeck Distinguished Professor in Advanced Imaging and Vice Chair for Research in the Department of Radiology and Biomedical Imaging at the University of California, San Francisco. She is the co-director of the Center for Intelligent Imaging at UCSF, a fellow of the American Institute of Medical and Biological Engineers, and a fellow of the International Society of Magnetic Resonance in Medicine, which she was recently awarded the ISMRM Gold Medal. And today, Sharmila will be talking to us about some translational AI methods for musculoskeletal imaging. Sharmila, the floor is yours.
Thank you so much for the introduction. I hope you can see my screen and hear me.
Yes, it looks perfect.
Great, perfect. So at the Center for Intelligent Imaging at UCSF, our goal is to apply AI across the entire life cycle of medical imaging and start with technology development for image acquisition and reconstruction for faster, safer, quantitative imaging, move to actually doing tissue and organ characterization, normal and pathological feature extraction from images, and then ultimately validating these methodologies in real structure function prediction. And I'm going to use degenerative joints and osteoarthritis as a marker for today's talk. How we, in education and outreach forms a huge part of our efforts, and we always have students, postdocs across the board involved in many of these activities. The idea here is that even though we do each of these pillars, as you see here on the slide, each pillar interacts with the other. So the ultimate goal would be that we would be doing clinical, integrative tissue and organ characterization on the fly, potentially modifying the acquisitions, and we are taking the steps moving there. Let's talk a little bit about osteoarthritis, since I'm going to be using this as an example. It's a degenerative disease. It's widely prevalent, and by the year 2020, there are going to be a large number, 59 people who are going to be affected, 59 million people who are going to be affected by this disorder. It, of course, causes joint pain, and the ultimate outcome is joint replacement. And now younger and younger people are going in for joint replacements due to the loading of the skeleton, high activity rates, and many other reasons. There are demographic risk factors, and the patient-reported outcomes are typically pain, which are used to characterize this disease. The FDA only accepts radiographs which depict late-stage osteoarthritis and bony changes. Magnetic resonance imaging provides exquisite detail about the tissues involved in osteoarthritis, such as the cartilage, the meniscus, the bone, the inflammatory changes. But because the grading of these images, which are used in the research and in the epidemiological world, are not only tedious, but there's also a lack of standardization, reliability between different readers. It has never made it to clinical practice. So given that, our goal was to translate quantitative measurements of joint morphology to clinical practice. I'm going to use the knee joint as a model in this particular case and walk through that, identifying some of the issues. Clearly, one of the requirements is to identify the tissues such as the cartilage, meniscus, ligaments, and so on. And so you need accurate segmentation. I'm not going to go into the accurate segmentation details here because many of you have been partners with us in working on various challenges. We have a robust ensemble model that identifies these tissues of interest and in different compartments of the knee, for example, in the patella, femur, in different tissues, meniscus, cartilage, etc. Moving forward, what we wanted to establish is MRI grading after the segmentation of this knee joint so that we could automate or assist the radiologists in MRI gradings and actually bring forward the research and the other gradings into the clinical world. In order to do so, we basically had trained a model using multiple scanner acquisitions, using the osteoarthritis initiative and some of our research data sets, and developed a multi-compartment whole knee severity staging, which grades lesions in the cartilage, it grades lesions in the bone, ligaments, meniscus. And when you look at the different compartments and different loading and unloading regions of the knee, it actually generates 60 probabilities. The multi-class sensitivity of these models ranged from 70 to 89% for cartilage, higher for other tissues, but different for meniscus, bone, and ligament. It was comparable and even in some cases better than the radiologist. But the question then arises, how do you put this into practice? And how do you present 60 probabilities to the radiologist? So we came up with this graphical reporting methodology, which looks at the various regions in the knee and the compartments and grades it based on the probability of there being a lesion and provides the confidence as well and the probabilities for the radiologist to see. Having said that, we had to test it out to see how the radiologists actually utilize these models. We did a study with 50 clinical MR cases graded by a senior radiologist and 2 trainees. They did this independently and blindly without AI support. If you look at this graph over here, in yellow is the standard grading and the agreement between the trainee and the attending. In yellow, you can see it's variable for trainee 1 and trainee 2, and it's actually improved when the trainees and the radiologists were actually given the inferences derived from this model, and they did a second round of AI-aided gradings. The trainees significantly improved their agreement with the experienced radiologist. Well, now we have to move forward to actually do this in the clinical setting and randomized trials. And so in order to do that, we have actually developed a method of trying to deploy these results, taking the data which comes from the PAC station or from the scanner, going into an inference service, which is using Clara Deploy as a partnership with NVIDIA. The inference is made and the data is sent back is stored in an XNAT database server, and it's also sent back to the PAC station for the radiologist's opinion, which then it goes back into the XNAT server for continuous learning. So that's on its way. But the reality is we all talked about the ground truth. So there was a suggestion that we use a clinical outcome as the ground truth. And in this particular case, I'm showing you in the osteoarthritis initiative of 5,000 cases, we looked at the occurrence of the first total knee replacement within 5 years. We used demographic features as well as image-based features to go into a training model. And we found that this model actually predicted total knee replacement better than x-rays, and including of demographic features actually improved the MRI predictions of total knee replacements. The unusual finding and the most exciting finding was that we also had subjects who ended up undergoing total knee replacements but did not have radiographic osteoarthritis, and we call them control subjects because of that particular reason. Sorry, those were not the controls. These were the subjects who didn't have radiographic osteoarthritis, but this was also predicted by the model. So there are features which it's picking up, which were not picked up originally seen in the radiographs or by the other features that were extracted from the demographics. Now, fast acquisition, we're going to hear some more about this, but in order to make this a reality in the clinic, MR imaging times have to be shorter. So we've been looking at task-based reconstructions where the image reconstruction as well as the task is done from end to end, and the optimization is done on the features of the task, such as segmentation. And we find even with a 25% case-based sampling, we get fairly good results in terms of our segmentation and looks very much like the full case-based sampling. But the real issue is when you have small features, which you have, such as this meniscal tear. This is from the fastMRI NeuroIPS of last year. This is the ground truth. And for the NeuroIPS competition, even the worst and the best case, best models were not able to pick up this minuscule lesion. So it's our goal to try and pick up the smallest features which are pathological. And to that end, just want to present some of our latest results, which utilizes an attacker network in the training, which basically causes small perturbations in the signal, and using these perturbations and tuning the loss function so that we recreate these known artifacts, we've been able to reconstruct images where the smaller lesions have been detected. This was work done by one of the Berkeley undergraduates who works with us and was the best paper in MIDL some months ago. And we found that the number of lesions that were detected using this network were actually greater than that which was detected using the UNet. So all this is saying is that there are challenges in any one application, and there are multiple steps. And it is with this team and Valentina Pedoia and others in this team that we're heading to try and solve some of these challenges. And I just demonstrated it with this one example with knee osteoarthritis. Thank you.
Sharmila, for that great talk. It's really exciting to see how a host of different deep learning advances can really be integrated into the full clinical workflow. So I think that's really promising. So to keep moving ahead with our session, our next speaker that will be joining us is Dr. Shreyas Vasanawala. Dr. Vasanawala serves as the director of MRI at Stanford Hospital and Clinics and at the Stanford Children's Hospital. He also serves as the division chief of body MRI. He previously received his bachelor's degree in math from Caltech and then completed his medical training as well as PhD at Stanford. His research efforts are primarily focused on developing fast and quantitative MRI methods. And today he'll be talking to us about what Upstream AI is. So Shreyas, I'm excited to learn more.
Thank you very much, Akshay, and thanks to the organizers for this absolute pleasure to present here to this group. So I would like to talk about this concept of Upstream AI and describe what it is for the audience. When you think about medical imaging, there's a whole series of decisions or processes that have to happen in a certain order in order to reach a diagnosis. And so if you look at these steps, the first one is really just what imaging tests should you do? Which modality? Is it a CT, MR, X-ray, ultrasound, et cetera? Then the next question is, when should it happen and where should it happen? Often we have a large imaging system with many different off-site locations for scanning. And it's a question of where's the best place to put it for the most efficient scanning. The next question is, how should it be done? A lot of medical imaging tests are different from blood tests in the sense that you have to pick what types of imaging you want to do, even within a given modality. And there's a lot of different parameters that have to get selected in order to produce the optimal images. Next question is, who should do it? There's different levels of skill and expertise in people who are executing on these exams. And so it raises a question of how that should be done. Another question is, how should the images themselves be formed? There are different types of algorithms that can be used to form these images, and that leads to, again, a decision point in the imaging chain. Then there's a question of, once you have images, are they good enough? And if they're not good enough, are they fixable? Can we somehow improve their image quality so that diagnosis can still happen? And after that, there's once you have images, you then have a lot of options for image registration and segmentation. You've seen some examples of this earlier today. And then finally, there's the actual diagnostic question at hand, the image interpretation. So all of these areas that I'm showing in this light blue are areas where an image has still not yet been formed necessarily. And this is what I'll refer to as upstream, because it's upstream from the actual image formation itself. There's another aspect to this as well, and that is, how should the imaging equipment itself be designed and constructed, which also is in the realm of upstream from the actual image formation itself. Now, each of these is a source of potential error and delays in the diagnostic process. And so these all offer opportunities for AI to improve the process. I'm going to just highlight in this brief time a few options in some of these areas. I'll start off with the area of how images should be formed. So here's a picture of an MR scanner. This MR scanner is a sophisticated device that is essentially a sensor for electromagnetic signals. And so here you're seeing actually these signals that are distributed in the Fourier domain of an image. Now notice that this takes quite a bit of time to acquire this, and Sharmila alluded to this as well. These exams take quite a bit of time. And so once you have all of these, you can then perform an inversion. So it's a mathematical algorithm that leads to an image. So here's an example of a liver, and you can see the arrow pointing to a blood vessel in the liver. Now this image quality is pretty good, and you see the vessel quite clearly because we've sampled all of the Fourier data in order to form the image, again at the cost of a long scan. So an alternative is to just subsample. And so here instead, we can just take a few samples from this Fourier domain and still try to form an image. But when you do that, you see that the image is grainy and has some artifacts in it. And so that vessel that was seen earlier so clearly, it now has less clear delineation. There's a whole literature and a field to trying to reconstruct these images from this reduced amount of data. One of the interesting explosions that's happened recently, especially in the MRI community, is a host of deep learning algorithms that can now reconstruct good quality images. And so here I'm showing you on the right an image that has been reconstructed or formed with an AI algorithm. And so instead of looking at AI for diagnosis or analysis of images, it's now being used to actually form images. An example of upstream imaging. This is some nice work that was done by a graduate student at Stanford, Fei-Yu Chen. Here's another example from another graduate student at Stanford, Christopher Sandino. And here he's actually looking at trying to form cardiac images. And so just very quickly, if you look at the left image, you'll see if you actually just perform a conventional reconstruction where there's very little data to start with, you get images that have some sense that there's a beating heart there, but it's nowhere near adequate for diagnosis. Whereas the right algorithm that he developed shows the heart beating quite clearly. Now, this applies not just to MR, but to other modalities such as CT. And so this is some nascent work from Adam Wang and Bhavik Patel at Stanford in collaboration with Debashish Pal, who's with GE Healthcare. And the essential idea here is that with CT imaging, there is quite a substantial radiation dose. And that dose is conventionally required in order to produce images that are not so grainy, such as the middle image shown here. However, we'd like to minimize this radiation dose. And one of the interesting opportunities here is that the process by which CT images are acquired is that you start with some conventional projection X-rays, as shown in the left. And these X-rays contain patient-specific information that can be used to predict where radiosensitive organs are and to minimize the radiation dose to these organs while still maintaining an optimal image quality. So this is again an example of upstream AI in which we've not actually used AI to interpret images, but it's how to acquire them in the most dose efficient way. Now here's another example. This is from a graduate student, David Zhang. Here I'm showing you an MR image, and you can see that the image is quite blurry. These show some vessels in the liver that are blurred. And the reason why they're blurred is that in any MR scanner, the magnetic field is not precisely known and is not a completely homogeneous. And this leads to these artifacts. So his interesting work has looked at how do you actually predict what that field is and then correct images using again, a neural network. And so here's an example where you see that the images have been cleaned up quite nicely. Now, here's an interesting example from that same algorithm that he's developed. It was never trained on pelvic MRI data. And you can see from the black arrows that images that were acquired with a very long scan and a more resistant acquisition to field inhomogeneity shows this blood vessel very sharply. However, if you scan faster, where you're more sensitive to those field variations, you see the vessel is blurred. You see another vessel here with this dashed arrow. And then after running it through this algorithm, you see that actually these vessels are now quite well delineated. And this is off of a type of data that the algorithm had never seen before, a nice example of generalization. Now, here's another example of Upstream AI. So here's the question of who should do these exams or how they're going to be done. And so this is an example from Hart Vista in which the MR scanner is getting a cardiac MR, and it's being done without any person actually touching the MR scanner during this acquisition. So all of these complex planes that have to be planned out to determine standard views of the heart are all happening automatically without a user interacting with the scanner. The nice advantage of this is that you wind up getting very consistent views, and also the process of acquiring them is much faster since an individual doesn't have to take the time to plan them out. And so in this short time, I've shown you a number of examples of upstream imaging. One example that I haven't shown you is just even how to schedule the exams, and can you use deep learning types of approaches to actually get higher throughput on your scanners and thereby reduce cost. Once you have these exams scheduled, you can then use deep learning approaches to try to reconstruct better images from less data, and then actually identify artifacts that may be present in these images and determine whether you can wind up fixing them. And so I hope I've convinced you in just a short time that this area of upstream AI, prior to having any images for diagnosis, represents a tremendous opportunity in medical imaging. Thank you very much. This is just a quick picture of some of the collaborators on a lot of this work, including folks at UC Berkeley and GE Healthcare. Thank you.
Thank you, Shreyas, for that wonderful overview of just the whole host of problems that we can really try to address with deep learning in the entire radiology workflow. And I look forward to seeing more advances there. Moving along with the session, and I think what's a very relevant talk based on MRI reconstruction that was pointed out in the last couple of talks, we'll be having our next speaker, Nafisa Yakubova, who will be speaking to us about machine learning for medical imaging, the fastMRI case study. Nafisa is a program manager at Facebook AI Research, and her broad research interests focus on the intersection of AI and biomedical sciences, as well as social good applications, such as the fastMRI, which she'll be speaking about, as well as COVID-19 clinical prediction, modeling of RNA sequences for designing new drug treatment strategies. Prior to Facebook, Nafisa came from a medical background, medical and a tech background, and she consulted and supported biotech as well as digital health startups. So I'm very excited to hear about fastMRI, Nafisa.
Thank you. So fastMRI is one of our projects where we use AI to accelerate MRI acquisition. And if you've ever had an MRI scan, you have to go into this machine and stay perfectly still, and it generates beautiful looking images of our inner parts. But it can be slow, like the other speakers have mentioned. It can take any time between 20 minutes to an hour. And if some people are just too sick to remain still for long periods of time, it's really hard for children as well. And there are some cases where you would ideally want to use MRI, but you end up using CT or X-ray images because they're faster. But if we could use MRIs for those cases, you could also reduce the exposure to radiation. That is also a matter of moving organs where you have always beating heart and lungs that introduce motion artifacts to the image, and you have to hold your breath. So of course there's like a big need to speed up MRIs. So between going into the scanner and getting the image, there is a part where you're acquiring measurements in frequency domain that's called K-space, that you're sampling each line one at a time, and the scan time is proportional to the number of measurements you're getting. And once you have this K-space, it's converted to the image that radiologists and patients are used to seeing. So in the upper part here, you see fully sampled case space generating a beautiful image of a knee MRI. And if you undersample, meaning remove some of the lines, it's already creating artifacts. So the question we're trying to solve with fastMRI is, can we use machine learning to create images that look like the original, but still using undersampled data? And this is a very interesting AI problem because you've seen recently how computer vision field have progressed. Like, for example, you can generate the impressive looking images of the celebrity faces, right? And we start to think, what are the applications that can benefit society in this space. And medical imaging is a great cause. However, it comes with challenges of its own. You first of all need a data set that is large enough so you don't end up overfitting your models, and also the same data set that other researchers could ideally access for the sake of scientific reproducibility. And evaluation metrics that work for natural images don't always work for medical imaging. And you, of course, have to come up with algorithms. And here I say algorithms because there could be more than one solution for this problem. Of course, the biggest risk in MRI image reconstruction is you do not want to introduce information that wasn't there or remove something that's supposed to be there. And we have a group of researchers looking into increasing confidence in the reconstructing images by creating error bounds for each pixel, and you can learn more about it as well. So here you see the image of a knee where pathology is just a few pixels large, and if you over smooth it, you're missing the whole point of having the image reconstructed. So it's a really difficult problem. And for this, we have teamed up with NYU School of Medicine, because complementary expertise is really at the core when you're working in machine learning space and medical imaging. And luckily, we both our organizations shared the open scientific research philosophy. So even before we started building solutions in-house, we started sharing everything with the rest of the world. NYU has generously made their data set public, and it contains thousands of knee and brain images. And it is the largest case-based data set. You might have seen data sets publicly available that contain MRI images, but it is rare to find MRI data sets in the raw format of a frequency domain. We have also early on decided we want to focus on benchmarking, having open source code and models to host public challenges and really focus on evaluation metrics, and make sure that we have MRI physics informed models and solve together with radiologists at every step, and really focus beyond paper, like can we validate in the clinic eventually, and will it be used in the real world? And here you see 4 times undersampled data acceleration. One is a reconstructed image using deep learning methods. Another is the original ground truth. And they both seem to have, like when in reconstructions, seems like you can still have the key information that you need to focus without altering it. And currently we are doing clinical studies to validate this at a systematic level. In addition to this, we've also looked into active acquisition. So how do you know which case space lines you should be undersampling? And because they're not all created equal. So a group of scientists in our team have also looked into creating a loop of learning. So it's more like a personalized imaging for the future if you can tailor sampling patterns for each individual and each case. So just to wrap up, because time is short, is that the key lessons here has been combining MRI domain knowledge was really essential for us in designing better deep learning methods and modifying them, and really focusing on clinical validation has been the key. We currently have retrospective and prospective studies running at NYU. Hopefully we can share the results with everyone soon. And we also have been looking into, in addition to MSK cases, we look into creating brain MRIs, maybe at even higher accelerations, and looking into dynamic MRIs where we have moving organs, and focus on vendor testing. So because everything is open source, we are giving away all the models we are developing. So MRI manufacturers and other research groups can take it and incorporate. So we have to learn if it's going to work in all kinds of scanner types and cases. So this is a part of the, like the end goal to make sure it gets to the real world. And in the spirit of open scientific research, we are hosting another fastMRI image reconstruction challenge this year. Last year, we focused on knee MRIs. This year, we're going to focus on brain imaging, looking for 4 and 8 times accelerations, and also transfer track, where we use different scanners, not only the same type, to make sure that it generalizes. And the challenge opens on October 1st. But you can go to fastMRI.org website right now and access the data set and the leaderboards and start, being part of this experience. We also have other projects in machine learning and clinical space, such as COVID-19 predictions. I don't have time to go over that, but if you visit ai@facebook.com website, you can learn more about our other projects. So with that, thank you.
Thanks for the very nice talk, Nafisa. And I definitely have to applaud your and your team's efforts in making all these data sets publicly available. I feel like that can really benefit not just the medical imaging community, but having other people outside of the medical imaging community access that data can definitely help drive improvements.
Yeah, thank you.
And now for last but not definitely the least speaker for our session, we'll be having Dr. Jayashree Kalpathy-Kramer speaking to us. Dr. Jayashree Kalpathy-Kramer is a director of the Quantitative Translational Imaging in Medicine Lab and the Center for Machine Learning at the Athunala Martino Center for Biomedical Imaging. She is an associate professor of radiology at Massachusetts General Hospital and the Harvard Med School. She's the scientific director at MGH and Brigham Center for Clinical Data Science, and also a senior scientist at the American College of Radiology Data Science Institute. The work that she does in her lab spans a spectrum from novel algorithm development as well as clinical deployment. And today she'll be speaking to us about collaborative learning, improving model robustness without the need for data sharing. Jayashree.
Thank you so much for the invitation to speak here. It's been an absolutely fabulous event. So the next few minutes, I'm going to just discuss some of the approaches that we've been taking to collaborative learning. As you've heard, may have heard earlier in the day, there's been a lot of interest in these approaches. The basic idea with some of these methods is that you're allowing the models to learn from disparate data sources, perhaps at different institutions, without necessarily having to share the data. You've also heard all day thus far and continue to hear how deep learning is revolutionizing medical imaging along the entire spectrum from image acquisition to detection, prognosis, and so on. And thanks to a lot of the advances in the community and availability of data and so on, it's getting increasingly easier to develop algorithms. You may have seen some of the phenomenal work from Pierce Keen and others at Moorfields, where their clinicians created algorithms without necessarily the need for coding. Despite all of these advances, it can be quite challenging to create algorithms that are very robust, fair, unbiased, can tell you when they're wrong, and things like that. So that is very much still a work in progress. As you've heard, the data set sizes that we tend to be much, much smaller in medical imaging compared to what the computer vision community has been used to. Perhaps as a consequence of some of that, the models that we tend to be somewhat brittle. There's not as much work that we'd like to see in the external validation and generalization of these models that we'd like to see in the published literature. Very often people are still evaluating on the same kinds of internal data sets that models are trained on. As most people are quite aware, these models don't perform nearly as well on data that's quite different than the training data. There's different ways in which data heterogeneity can manifest itself. So you might have different institutions that have different mix of the prevalence of the disease. Sample sizes might be different. The scanners might be different. You can also have different populations, different annotations. A lot of this heterogeneity leads to somewhat reduced model performance. Here is an example from the BEMIS trial, the work that we did with the ACR. This was data looking at just a fairly straightforward task, looking at breast density classification. If you don't really think about the images themselves and just use sort of pi diagram to convert it without setting up the process properly, you'll see a complete mishmash of images that make no sense whatsoever. If you then start to look at how to interpret the images correctly, but look at the histograms, you see that these scanners produce very different types of images. So if you train a model with scanner 1 and test it on scanner 1, you get very good performance, and you see that on the on-diagonal elements. But the minute you take a model trained on scanner 3 and test it on scanner 1, you'll see that the performance degrades tremendously. Additionally, you'll see that the performance of the models is quite different on the different sub-races. So it really becomes quite important that we think about how to generalize these models. One way to do that is to have a nice central repository, and there's many wonderful examples of that. Stanford, of course, has been phenomenal in their generosity in making a lot of these data sets available, as has organizations such as RSNA. These are great, of course. You get multiple institutional data. You can really dive into it. And what we see, of course, when you do that is that a model built using all of these data sets that for the breast density case performs well when tested on all of these data sets as well. So this definitely is one way to mitigate this problem of the lack of performance on external data sets. There's a number of challenges with that, though. So you need a lot of resources for de-identification. There's concerns in some institutions about making data publicly available. There's a lot of IP around it. There's technical challenges to that as well. So there's a variety of collaborative learning approaches that have been proposed. So there's model ensembling, sort of the most trivial one, federated learning, cyclical weight transfer, split learning. There are essentially different technical ways to achieve the same end goal, which is to create a model that has essentially seen all of the data without having to share the data itself. So federated learning is a very popular approach, and it's typically accomplished using either weight averaging or gradient averaging. And the basic algorithm looks something like this. You have a parameter server that's a central server. Each institution has local training that happens. You initialize the task. The model's trained locally at each institution. You upload the weights to the central server. The models are aggregated, and this process continues till you get some kind of saturation. We did this for real in a case with breast density, again, with working with NVIDIA and ACR and Stanford and many other institutions, where we showed that by doing, you can see that the data look quite different from the different scanners. Despite that, doing this federated learning approach, we can get a pretty good model, improve the performance over the local models, as well as have a pretty good central model. Another approach that Dr. Rubin and I and our groups have been working on is cyclical weight transfer, where the idea is, again, you sort of continue to train the model at each institution, pass the model on to the next one, and continue to improve performance. And we've shown that this approach can allow you to get pretty good performance on the central, similar to the centrally hosted data as well. This can be really important in cases of rare diseases where you may not have enough data at a single institution to get a good model, but by doing this over multiple institutions, you can get pretty good model performance. Another approach is split learning, where essentially the front of the model and the back of the model are the local server, but the middle part of the model is on the central server. And this is yet another technical approach to do all of this. So there are many, many technical ways to do this process, but this even more challenging is the logistical considerations. So you need to have a consortium of willing participants, have agreement on what the clinical data is, how you're going to annotate it. So standards around all of that are quite important. The infrastructure to enable secure communication between sites really need to decide on IP and publication strategy upfront. In terms of technical challenges, there's again many different ways to do it. Dealing with the data heterogeneity is an important one. Where some of these methods are more prone to things like catastrophic forgetting, and how do you deal with that? Number of security and privacy considerations as well. So just to summarize, a lot of these techniques have a lot of promise. However, the models can be quite brittle, and so we need to think about collaborative learning approaches to improve model performance. With that, I'd like to thank our group. And there's a number of resources out there in terms of both software as well as publications in this area, and frameworks such as MONAI that might also be useful in this space. Thank you.
Thank you for that very nice summary. I feel like being able to aggregate large data sources from multiple different hospitals is really what's lacking right now to be able to build generalizable and robust models. So I'm really excited to see your work there. So I think right now we'll be transitioning to our panel session where we'll just aggregate all the speakers that we've had so far. And I have a few questions, but I look forward to the audience submitting more questions. So I wanted to start off with a concept that had come up quite a bit in multiple different talks. It's this notion of building generative neural networks, for tasks such as super resolution, MR reconstruction. All of these problems are fundamentally ill-posed, where you can have multiple different solutions. So is there any way that we can try to ensure that we're not hallucinating features that may not be there, especially when we're thinking about deploying this in a clinical perspective? So I'd love to hear any insights that people may have in that regard.
So I can certainly talk about that. it's something we've looked at a lot, and we have that question every time we present this work over the last 2 or 3 years, in fact. And we've never actually seen it come up. So the first thing to mention is it seems much more of a speculative concern than a real concern. But really the key thing to do is the thing that I showed in my talk, which is to actually show the results of using a downstream task with a generative model. So for example, in our case, we actually had some experts on synaptic vesicle detection look at the images and circle the synaptic vesicles and compare the error rates, false positives and false negatives between the various choices, as well as the original high resolution image. Because in the end, you're doing the generative model for a reason. So you can actually test that downstream task, that reason you were doing it in the first place.
And maybe it'd be great to get Sharmila's or Nafisa's perspectives here, because you'd shown a few examples where there's small pathologies that exist in images. What has your experience been about maybe the false negative or even the false positive problems that have been brought up?
So a lot of times when we've had false positive or false negative rates, basically from the models, and have gone back to the radiologists for verification, particularly in the knee, for example, it often has been that the radiologist post hoc, after looking at the AI-assisted grading, has agreed with the model. In cases where, for example, in one case, the meniscus was completely misgraded, was because there was no meniscus. It was so macerated that there wasn't even that feature available to grade. So under those circumstances, the radiologist would said, I'm not grading this, but the model went ahead and graded it because it expected something to be there. So from that perspective, I think that once you actually go and deploy some of these models into into the clinical workflow and get the feedback, and it's used as a decision support tool with all the other history, the electronic health records, I think we will learn more from it. But we have such a variability in the ground truth that we're using as labels that I shouldn't say most of the time, but many of the times we found the radiologist says, oh yeah, I see it now. And so that has been our experience in the knee. And the features are really small in the knee, as you know. And so we've had that. That's been our experience in the knee.
Nafisa, do you have anything to add?
Yeah, I can add to that. We have several things. There's no perfect solution for it, right? So we have a fully sampled case base, and we retrospectively go back and undersample. So we have a ground truth to compare. In our experience, I think it's performing pretty well so far. Of course, we're conducting double-blind clinical studies where quantitative metrics is not enough, just asking radiologists is not enough. So we are doing a combination of the two. And at a certain acceleration rates, it starts having that problem more often than lower acceleration rates. And so you go to radiologists, ask them, okay, can you find that diagnostic feature they're looking for in this image or not? And there's always a ground truth from fully sampled you can go back to. That's a retrospective one. We're also looking into some of the patients, not as part of our study, but naturally they go into surgeries, right? So you have even more ground truth than radiology readings. You can actually go look inside the knee and see if there's been a damage to meniscus or something else. So those things could be part of the study. So I think that that's part of the solution so far, because there is no ideal evaluation metric for it. We have to combine those methods.
Yeah, and it's really interesting, similar to what Sharmila mentioned too, that there may be disagreement within the same person if they look at the same case on different days, or maybe if multiple different radiologists are evaluating the same image. There's no perfect ground truth. And Joshrey, I wonder how that ties into this notion of federated learning, where, since we deal with discordance between readers, do you see that being a challenge for federated learning, where there's discordance between how different hospitals actually perform their classifications?
It's a great question. It's something that we've actually studied somewhat at length. So we've done a number of studies, even within an institution, having multiple people rate the same images. And what we see is quite a spectrum in ratings. People tend to be biased, is what we've seen, and especially when you're having something on a severity scale. And so somebody's 2 are very often somebody else's 3. And we see this sort of fairly systematic bias. And we actually know how to learn those offsets between sort of what the median should be and what the each person might be, but it really is very much more of a challenge when you're doing this in a federated system. If there's no consistency between the rating protocols or the annotations, that makes it a lot more challenge. On the other hand, there are methods that you can actually learn, almost like an EM type of way. You can learn the difference between the predicted version versus the person's version and learn the offset between those two. So those are things that we've looked at, but it's, at least in our experience, it's very much a challenge. And we see that with the data set we got with the multi-institutional trial, clinical trial data, we can see clear differences between almost like the model's output expected performance and what each site rated at the same image as.
In the interest of time, I had a quick question for Shreyas, and this has actually been brought up by one of our audience members, Sandy Napel, who's asking, would it be better to actually fully integrate the upstream and the downstream AI techniques into a single stream as opposed to keeping them separate? Do you have any thoughts about that, Shreyas?
Absolutely. And I know multiple people have explored in this area, and you can definitely achieve certain aims of, say, faster imaging speeds if you have a particular diagnostic endpoint in mind. So there's definitely value to that. And just to quickly touch on the previous question that you'd asked about regarding risks of hallucination, there's in some sense a little bit of a false dichotomy, because conventional imaging methods don't necessarily image the underlying object with perfect fidelity to begin with. So it's sort of setting the bar in a way a little bit higher than you would ordinarily. But that said, at least in the fields of image reconstruction, a lot of the approaches have network architectures that enforce a data consistency to begin with, and that reduces the risks of hallucination. And then, another aspect to this that I think is quite interesting is really looking at metrics of uncertainty in your image reconstructions. And for instance, being able to have minor variations to the input side of data, and then seeing, okay, well, what's the variation on the output side, is giving some metric of the, the uncertainty that you may have.
Yeah, that's a really good point that you brought up, that instead of just using only physics-based models or using only deep learning-based models, there's probably a home for combining both of these. So again, I would like to thank all the speakers for their fantastic talks and this really nice discussion that we had. I feel like this was very fruitful. Thank you again to all the speakers. This was great having you.